First of all, want to thank the organizers of the conference to invite me here. I'm very excited to be part of this. Really is part of this very esteemed group of people. And also very excited to share this case study today with you all, because this story that I'm going to tell you today is very close to my heart. It's just not work. It's a life mission. So the key question that I think about is this one, which is how can we improve access to life-saving vaccines so that we can prevent preventable disease? There are millions of people today that do not have the basic vaccines that are required to survive disabling diseases that don't even exist in this part of the world. A lot of work has been done, and if you think about how vaccines are produced, you can start to tease out where the problems are. A lot of the R&D work in terms of creating vaccines is being done in the West and in the developed countries. So all the innovation and discoveries of new vaccines is really only conducted in those countries where there are a lot of resources and the trained personnel. Manufacturing of vaccines is really localized amongst 5 major manufacturers of vaccines in the world: Sanofi, GSK, Novartis, Merck, and Pfizer/Wyeth. These companies have the biggest facilities in the world that create majority of the vaccines. And then there are a few low-cost manufacturers of vaccines in China and India. And when you think about where the distribution of the vaccines is occurring, most of these multinationals are providing vaccines to the West or to those parts of the world where the populations can afford those vaccines. And then the low-cost manufacturers that exist in India and in China sell their vaccines to the WHO/UNICEF, who then distribute these vaccines to the poorer nations. So there are some success stories in terms of improving access to vaccines through the UNICEF type of agencies who buy these vaccines from the low-cost manufacturers and supply them to the poor nations. So it's not that this problem is not being looked at. A lot of very smart people and people with a lot of good intent actually are thinking about this, and there are some success stories. For example, the pediatric vaccines, which are the measles, mumps, rubella, polio, DTP, diphtheria, tetanus, pertussis, hepatitis B. Some of those problems are to some extent being addressed reasonably well. But there are many that still go without basic vaccinations, and then there are some newer problems. And these newer problems require a newer type of solution. So what are these problems? I mean, 2 of them have occurred in the last 5 years. We've seen influenza pandemics. And if you think about the influenza pandemics from last year, the H1N1, the problem becomes very apparent. And the problem here was not that there were no agencies like the WHO or UNICEF to supply these vaccines. Those agencies still existed. But what became very apparent was that the distribution and manufacturing of these vaccines was really controlled by political borders and economic factors. In the early part of the pandemic last year, when people really didn't understand or didn't know what the extent of this pandemic was, the only countries that had access to vaccine were countries that either had manufacturing facilities in their own borders, so there was a political power, or those countries that had the biggest dollar, the biggest price that they can give to buy the capacity. And the analysis was very simple. The top richest countries, 10 richest countries, had access to all of the vaccine. So you can basically throw away this concept of having a centralized arbitrator of supply and demand of vaccine. There's no such thing. At the end of the day, when you have a crisis, it's the political and economic factors that determine who gets what. To me, that's a troubling thought. Because if somebody is born in a poor country, they're destined to die in a situation like this. And that shouldn't be the case if it is in our power to address it. And today, my humble presentation is a step towards that and a vision towards how we can address it with the kind of intellectual capability that all of you have here, which is all related to engineering. The second problem that is related to this is that you are seeing sporadic emergence of new pathogens that are locally that are local to certain geographies. For example, how many of you here have heard of this new virus called chikungunya? A lot of you don't know what this thing is. That's because we don't see this here. But it's out there. My dad, who lives and works in India, was contracted with this virus. It's a disabling virus that is a class of viruses called flaviviruses, and it affects the joints. It creates an incredible pain in the joints. And this virus is spreading, or has been spreading in India and other parts of the world like that. But we don't even think about it because it is not affecting our lives. EV71, how many of you know EV71? This is an enterovirus that is similar to the foot and mouth virus, and it causes a diarrhea type disease, and it's a big problem in the Far East, countries like Taiwan. And then, of course, there are more prominent viruses like dengue and malaria, which people know more about because they're well publicized. But the point is that there are a lot of infectious diseases that are out there that are not being looked at carefully because they don't affect us. And ultimately, I believe the only solution that we can have to address these local diseases or to really address the inequities in vaccine production and distribution is to provide control, both from manufacturing and from a development standpoint, in individual countries. And that's a problem, I believe, that is solvable with innovations in manufacturing. And innovations in local manufacturing. But that's a tall order, because you've seen talks before me that have talked about incredibly complex facilities for making vaccines that cost hundreds of millions of dollars and may take 5 or 6 years to build and require extremely trained personnel. If you're going to transcend that, you have to look at this problem differently. And I think one of the speakers prior to this talk talked about the concept of single-use manufacturing, which is, I think, a major force behind this. If you look at what is going to be needed if you are going to transport manufacturing, create what is called distributed manufacturing, you're going to have these factors to consider. You have to simplify everything significantly, because you're going to deal with people who have no idea what they're talking about. And you have to transcend infrastructure. You have to get away from incredibly complex utility infrastructures, which ultimately leads to this idea of single-use manufacturing. You have to cut down capital cost and commissioning time, but at the same time ensure quality, because what we don't want to do is to cut corners and create vaccines that are adulterated, because we don't want to create a new problem by solving one. And lastly, you want to create these facilities that can be used by multiple different products. So all of these things, if these are the factors that you need to be successful, there's one approach that you can apply, which is to create a vaccine technology that is a platform technology that can create multiple different types of vaccines using the basic structure or idea. And I'm going to talk about one of those. You have to start thinking about doing single-use manufacturing so you don't have to worry about cleaning and sterilizing the equipment. and then have modular facilities that can essentially be transported. One limiting case of this, which is something that is now in a prototype, is something that GE Healthcare, one of our partners, is working on, which is like a huge a tanker trailer that is a factory by and of itself on the wheels. That's a little bit far down there, but I think a modular facility is not such a difficult concept because this is being done right now. So I think applying these three things in a smart way can start to make some dent in this problem. And today, what I want to show you is our effort in that direction. So for those of you who don't know Novavax is a small company. It's based in Rockville, Maryland. We have about 90 employees. It's a publicly traded company. Its mission is to create virus-like particle-based vaccines. And I'm going to show you the idea of virus-like particles. And when I say its mission, ultimately, if you think about what we really want to do, is to really address that big picture question, is to really create a solution to the big problems in global health, but at the same time using technology and by maintaining the principles of capitalism, because we are not a charity and we want to make a profit. But we want to make a profit, but at the same time help the people at large. So this concept of virus-like particles is very simple. This is an example of the influenza virus. This influenza virus is made up of proteins and nucleic acids and lipids. And it's constructed when 8 gene segments of the influenza virus are expressed. When all those 8 gene segments are expressed, this virus is created. To create a virus-like particle, and the concept of a virus-like particle is really think of a decoy of the virus that looks like the virus, feels like the virus, appears like the virus to the immune system, but is devoid of the genetic material and some of the other proteins that are required for the virus to grow. So if you can create the decoy, and we can do that by expressing the surface proteins of the virus and having a scaffold protein. So the scaffold protein is the matrix proteins, which creates a structure, and then the surface proteins, the hemagglutinin and neuraminidase, HA and NA, which are basically the sensor proteins that show to the immune system that something new is around. And so that's where the immune response comes from. Those are the 2 immunogenic proteins. So if you can take these 3 proteins, and our idea is that if you express these 3 proteins together, these 3 proteins will self-assemble just like the virus self-assembles itself and create these particles. And that's the idea. So we take these 3 proteins and genetically engineer them in a system called baculovirus. And I don't have to go into those technical details, but the idea is very simple. You basically put them in a virus which acts as a vector, goes into a cell, takes the cell machinery, these 3 genes are expressed, these 3 proteins self-assemble, and these particles spit out, which we can purify and use as vaccines. And because these particles are extremely similar in size and shape and in the outside decorations to the virus, they are very authentic to the immune system because they appear like it's a natural infection. And that's why they result in a very robust immune response. But because these particles are not able to replicate, they cannot cause disease and overwhelm the system, and therefore they are safe. So you get the best of both worlds. You get a safe product, but you get a very authentic immune response. And this is an example of a virus-like particle that is seen through an electron micrograph. And you can see that the virus particles have, the outside spikes of the spike proteins of HA and NA, which look very similar to the actual virus. This picture actually happens to be a virus-like particle of the 1918 Spanish flu virus, which is interesting enough because that's a whole story in of itself, which I'll talk to you about later. The manufacturing process for making these VLPs, again, is very simple because, as I said, we are using single-use equipment. And one of the reasons why we can use single-use equipment in this example is because this virus is not pathogenic, and so you can use plastic or relatively you don't have to use stainless steel and have all kinds of constraints on the system because of you don't have to worry about it getting spilled or broken and causing harm to the people who are working on the thing. So you have much more leverage in terms of using these type of single-use equipment to produce the virus-like particles because you're using rather innocuous materials. The idea is that we have a number of different equipment that we have been able to get and then connect them in order to have an end-to-end solution that is essentially portable. None of these pieces of equipment here cannot be shipped from one place to the other. The other thing is that none of these pieces of equipment rely on the infrastructure. That is, in the factory in which it is produced, there's nothing there that is dependent on that factory. All of this equipment is somewhat, you can put them here, all this equipment, and you can produce the vaccine in this room. And I don't think you have to take a compromise on quality because all of this is closed equipment and it's all pre-sterilized. So you're not exposing the product to the atmosphere. I'll just give you a couple of examples of where how we scale this. This, right now, the reactor is a small one, but you can go to a bigger one. I'm sure some of the previous speakers have talked about these type of systems. Again, the interesting part of this is that this looks like a stainless steel tank, but it's a cutout from the top. And all you've done is you've put a bag inside there, a plastic liner, and you put the media and you grow the cells and produce the product in this. And once you're finished, you can pull your product out and throw away the bag and put a new bag in. It's like a garbage bag that you can just put in there. So it's a very simple idea. And again, these bags and this equipment can be transported anywhere in the world. On the downstream side, which is how to purify the product, you don't have to worry about using incredibly large columns of chromatography. This is an example of you can have these small pre-packed columns that can be used to process very significant amounts of material because they take advantage of this valve array that opens and closes to continuously go through a series of these columns. You can purify the product at the same time. As one column is finished, it starts to clean. So these valves go back and forth in terms of the cleaning and the processing mode. And by doing that in a smart way through using engineering, as in this example, you can have what would require a significant footprint and significant size equipment to a very small tabletop equipment and process very large quantities of material. So that's the idea. And if you then pull it all together and think about how do we use this in the case of a pandemic, you have the recombinant technology, which, as opposed to the egg technology, can reduce the time to create a new vaccine from 6 months to 3 months, approximately. And then if you have the cell culture system to produce the product in these bags of media, you can produce a large quantity of product very quickly so that you don't have to worry about product dribbling out at a slow rate as we saw last year in the US. So you can have this surge and you can protect your people before the second wave of a pandemic. So that's the idea of this technology. So this is all theoretical. So what did the company do last year? Remember, we are not FDA approved yet of our VLP vaccine. So we are somewhat constrained. So we are in the middle of a pandemic, not an FDA approved product. How do we respond? What do we do? What do we do to help people? So first thing we did is that we got the gene sequences from the CDC on April 24th, which is about 6 days after the first news of this pandemic came about. The CDC published its gene sequences of this new virus, the 2009 California virus, which they thought was causing the pandemic, H1N1. We got the gene sequence, and within 4 weeks, we were able to provide the first prototype of a VLP vaccine to the CDC. The CDC took this vaccine and did an experiment in ferrets, which is the appropriate model for influenza, animal model for influenza. And they were able to show that a single dose of 15 micrograms of this vaccine was sufficient to protect those ferrets. And that gave them a lot of confidence to tell the rest of the manufacturers, the big manufacturers who supplied the vaccine to the entire world, that one shot of 15 micrograms of a hemagglutinin-based flu vaccine will be enough to protect people. And it was borne out over time with clinical trials. So we served the public agencies, public health agencies, by providing the first vaccine to them so that they could do this fair study. It was not GSK, it was not Sanofi, it was not Merck, it was little Novavax that did that. The second thing we showed was that within 11 weeks of the emergence of this strain, we were able to produce a GMP, what is called Good Manufacturing Practices, CGMP, Current Good Manufacturing Practices based product, which could be used in humans. And that was used not in the United States because the US government was interested only in procuring vaccine from the established manufacturers that were supplying seasonal flu vaccine. We were able to convince the Mexican authorities, which was the eye of this pandemic, eye of the storm. This is where the pandemic started. And in this country, there was no vaccine. Nobody was supplying vaccines. It was not the top 10 country in terms of economics or political power. So here's the place where there's no vaccine, and this is where the pandemic started. And we were able to convince the authorities in Mexico that we could supply them vaccine so that at least they could do a clinical trial in their country. This vaccine was produced in our facilities, which I'm going to show you what kind of facility we have, but it uses the single-use equipment, and we were able to provide this vaccine to the Mexican authorities, and these are the data that came out of that vaccine study. And this is a dense slide. I'm going to walk you through this very carefully, but let me tell you what we did. On April 24th, we got the gene sequences. On October 19th, we started this clinical trial. On December 2nd, 2009, we had these data. And what these data, big picture, tell you is that this vaccine is well tolerated and it is highly effective. There are three things that the FDA or any regulatory authority looks at when it looks at efficacy of a vaccine, of at least a flu vaccine. What is called a fourfold rise. Fourfold rise is the antibody titers that you have against hemagglutinin, which protect you. You have, say, X, you need to be at 4X, 4 times the baseline. So how many people who got the vaccine got a fourfold rise in their antibody response? And you can see that there's a dose response between 5 to 45 microgram of dose, going from 43% to 69%. Of course, there was some background infection that was going on, of course, in the middle of a pandemic, so 4% of the placebo also got that rise. The second thing that look at is how many of those people have a titer above a baseline, which is considered protective. And let's call that number 40. In the jargon of flu, it's greater than 40. 80% at 5 microgram, 89% at 15, and 90% at 45. There was a lot of background infection, so about 38% of the people who had placebo also had that, basically from natural infection. And then GMT is the Jomet mean titer of the antibody responses, and you can see that these numbers are significantly higher than what is in the placebo arm. So when you look at these data, the DSMB, or the Data Safety Monitoring Board, the overseeing body that was looking after this clinical trial, which is made up of an independent member of key opinion leaders of influenza in the United States, they recommended that 15 microgram was the right dose and one single dose was enough, which was what was predicted from our ferret experiments. So far, we finished a trial now of 4,500 people in Mexico, where we have looked at a dose response in 1,000 people. We've selected the 15 microgram, and then we've taken the 15 microgram dose and given to another 3,500 people. So now we feel, and there are no safety issues whatsoever. So what we've shown is that we took a risk, Mexico took a risk on us, and we showed that with this little company that we could create a vaccine and respond fast, and you don't have to rely on significant resources of mega companies, that you can do this in a company with less than 90 people. And if we had the resources, if we had the order, we could produce a significant amount of vaccine. What's the bottom line? Now we can get fast-track approval in Mexico, we can supply product, and these data are resonating globally. And we can suddenly become a player. And how did we become a player? Without the resource of the big companies, we became a player because we relied on a completely different paradigm of manufacturing. And that's the model that we can use for the rest of the world. This is the facility. It looks like a spaceship. It's got 4 rooms. 4 rooms that are clean rooms, class 100,000, that have electrical outlets. No stainless steel pipe spaghetti whatsoever you see here. And this is the facility in which we can make that product. And we believe that we can do this around the world. We can go to a different paradigm of manufacturing where we get away from economy of scale of a centralized facility, but we can go to the distributed manufacturing approach because you're taking the cost structure away from economy of scale. You're taking the cost structure away from the fixed cost. You're putting the cost structure into variable cost. And with variable cost, whether you make 5 million doses or you make 50 million doses, you do not take a penalty on cost of goods. And that's what makes this facility, this approach of distributed manufacturing possible. We have not just put it on paper in a concept sheet. We've actually applied this. This is a joint venture we've done with India, with a company in India called Cadila Pharmaceuticals. There's an agreement we signed on May 31st. We started doing the technology transfer on March 31st, we signed the agreement. On May 2009, we started doing the tech transfer. We broke ground on a facility in India in November of 2009, and next month we'll have the facility done. And by July, that facility will be operational. So we believe that not only have we created a vision, we have put this vision into reality. We have a lot more work to do, but I believe the future has a lot more to offer. We can take this vision to the next level. And the vision is, can we go and take this idea to a franchise model such that we can have a recipe book. We can have a secret sauce. Just like Coca-Cola or Dunkin' Donuts. If somebody wants to buy a franchise to create a vaccine, they buy the franchise from us. We give them the recipe book, and they can make exactly the same tasting donuts in other parts of the world that they make in New York City. And we give them the capability that they can then take that information to create vaccines that are locally important to them. So they can have a custom menu of products in different parts of the world. Thank you very much.
Madhu Raghavan from General Motors R&D. What will it take for you to go the next level and get the FDA approvals that you need to function here?
Right. So for the US, the FDA approval process is, there are two different regulatory pathways that we go. One is that you get a seasonal flu vaccine approved first, and then you can use that as your basis for getting the pandemic vaccines approved. We are likely going to take that approach because we want to have a commercially viable product. Pandemics come and go, and you never can predict when the next pandemic will be there. So what we are doing is, we have a separate program in addition to the pandemic influenza on seasonal influenza, which is an annual vaccination, and that program is going through the development process. We believe it's going to be in phase 3 soon, and once the phase 3 trials are finished, we will submit our file for FDA approval. Once that file is approved, then we can use that as the basis for then getting any pandemic vaccine approved. And that's the regulatory pathway that all the big manufacturers have used. That is the US pathway, but internationally we are going in a different way. We are having a separate file for pandemic, as we saw for Mexico.
Kevin Eberman from Medtronic. I have a question that kind of comes from my not knowing much about this probably, but so Ulrich talked about how there's a decision made each year that three types of virus go into the seasonal flu vaccine, and it's the same ones worldwide. And I kind of was thinking, well, that's kind of odd. I mean, is that necessarily a good thing? It's sort of like putting all your eggs in one basket. And what you're talking about, it seems to me, will create an opportunity for a different approach where you could locally have a different vaccine, even a seasonal vaccine that could be appropriate for the region, which I don't know if that's a good thing or a bad thing, and I'm just curious what your comments are on that.
I think that's a very, very good question, and absolutely, I think we've talked about this idea that why should there be one single set of viruses that should be used as a basis for creating a vaccine worldwide? Think about how this virus moves. The influenza virus starts, and this is published, by the way. It was a very nice paper somebody did. It emerges in the Far East, it goes through Europe, and then comes into the United States, North America, and then goes into South America and dies out. That's how the virus moves. So there is a lag between when the virus is seen in North America or Europe versus when it started in Far East. There is quite a good possibility that by the time the virus reaches North America, you have a different virus in Asia. So to your point, having a custom formulation for different parts of the world would make a lot of sense. Why that has not happened thus far is because the sophistication of some of the manufacturing is not there. The manufacturers are all sitting in Europe or they're sitting in North America. Very few companies have started to now build facilities in the Far East. And even those facilities are completely dependent on the epidemiology and the surveillance data of the CDC and of the WHO. And at the moment, they are not sophisticated enough to say that we are going to give separate recommendations to separate manufacturers. It is a very, very good goal, and this kind of an approach certainly can make that happen, because all you have to say is that you use different gene sequences to create a formulation in India or in China versus what you use in Europe. And you get away from this centralized laboratory situation which creates the seed for all of the manufacturers in the world. And so one of the problems that we have right now is the CDC and the WHO are the only two agencies that have the seed for every year. They supply the seed to all the manufacturers. It's completely centralized that way. But if you get away from the idea of a virus seed and you say all you have is a raw material which is genetic sequence, which can be emailed to you, well then there's no reason why you cannot have a custom formulation. So I think that question is right on mark, and certainly not very far from the future, but there will have to be certain policy changes in addition to some technological changes in order for that to really come to fruition. Thank you for asking.
Right here. Alex Yezerets, Cummins Engine Company. Admittedly, I'm very as far from the world of biotech and vaccine as it gets, but more of a question from just the economy of scale up and scale down. I mean, in addition to production itself, I would assume, especially in the process, which is that where the market is so sensitive, I mean, it's a human health, I would assume there would be also some component to economics of quality control, scale up and scale down, right? And that the franchise model you described would also require that, unlike Dunkin' Donuts or Kentucky Fried Chicken, right, you would have some sort of quality assurance and quality control, right? And so how do you envision in the distributed manufacturing model, in my superficial understanding, that becomes the key obstacle you would have to convince people of being able to overcome?
This is actually a very sophisticated question, so it's not at all naive. I think that is one of the reasons why this approach has not been widely used, is because these are the kinds of problems that are very distinct from the very simplistic idea of a Dunkin' Donuts model. It's because this is a very regulated industry. It is a health industry, and moreover, you're giving product to people who are not diseased. So it puts the bar significantly high. And for this thing to work, you have to centralize not only the production system, but also the quality control. So what does that mean? The quality control reagents have to be also coming from the secret sauce. So the centralized facility that is supplying the seeds or supplying the basic technology will also be supplying the quality control. And there has to be some sort of a synchronization of testing. So there has to be some sort of a lab-to-lab connectivity in terms of, whatever is tested in lab 1 is tested in the central lab. So as part of the tech transfer, there has to be analytical process or analytical test transfer as well. And in the validation of a particular franchisee to be ready to produce will have to not only pass through manufacturing quality attributes or manufacturing attributes in terms of yields, but also the quality attributes in terms of purity and identity, etc. So I think a lot of this has to be worked out, but as long as you have the vision and you can apply technology to work for you, I think the problem can be solved. You have to put the appropriate sandbox around it.
Kliagmachi, Princeton University. So the West has a heads up on which flu virus are coming each season, and we can do that. But how is the East, the Far Eastern, dealing with the emerging flu? Are there better strategies to quicker respond? And could we learn from it?
So in fact, there is a lot of effort and a lot of money has been put into surveillance labs in the Far East. For example, there is a lab in Hong Kong, which is probably one of the biggest surveillance labs, and it's funded by some of the folks within Hong Kong and also by the Gates and other type of philanthropic organizations that are creating incredible amount of surveillance networks in China. So they're on a daily basis getting about 100 samples from birds and other reservoirs of the virus, and they're sequencing this, and they're finding out what can grow in or what kind of attributes of these viruses are changing, and which ones can lead to a humanized virus. So there's a fairly large surveillance network that is now being put in place in Asia. So I think even the CDC and the WHO rely quite heavily on those type of surveillance networks.
Poyan Porbey Kepri. So again, from someone not in the field, if I understood you correctly, the way you were synthesizing that virus was you're basically building, if you will, the shell of this thing to emulate the virus and make the immune system think it's a virus to create the immune response. What sort of guarantee do you have that that model's always going to work? I mean, what happens if you miss one little substructure of that structure, and you get an immune response, but it's not the right immune response, and the virus comes and bam.
That's a good question, and we're not smart enough to understand the full immunology and the biology of these things. But we've been working on, for example, influenza for 50 years. So I think in case of influenza, the key determinants of what proteins are required for creating the right immune response is very well understood. So in the case of influenza, we know well as to what kind of proteins we have to decorate these particles with in order to get the right immune response. But your point is well taken, because in cases like dengue, which is an emerging virus for which there's no current vaccine, we have run into problems. Not we, but the field at large. And very interestingly, there are and I'll give you an example of dengue to illustrate the point that we don't understand biology very well yet. In the case of dengue, there are 4 serotypes of dengue. And if you get infected with one of them, you are even more susceptible to get worse disease if a different serotype comes and infects you. One would think that you will be immunized if you are infected, and you will be better protected. It goes the other way. So the vaccine strategy is also therefore has to be addressed, because you have to have the right balance of the 4 serotypes, so you're not going to cause problems in the system because you've mismatched the amounts for each one of them. So I think your point is well taken. Fortunately for influenza, we understand that enough that we can use this approach in a confident way, but it is not a slam dunk for everything.
Kaiser Max Planck Institute in Magdeburg. I have one question with regard to the downstream processing. You've shown us on slide 11 the bio SMB, you called it. I guess it's a simulated moving bed device.
Similar to, it's like a.
So what's the difference to a traditional carousel type lab scale stainless steel SMB?
I think that the big difference is that we can use disposable pre-packed columns in this, which we can use for one batch and then throw it away and just put another one. So this kind of takes you to the idea that you don't have to rely on infrastructure of cleaning and sterilization. You can have these pre-packed columns. You can put it in your SMB and process it. It's economical to take it and throw it away. And the other thing that it also does, I think that is true for even a permanent system of an SMB, is that the use of buffer, which happens to be very significant for large batches, can be cut down quite significantly. So again, you can use smaller facilities and produce the product that you need.
Gabi Breidel from the University of Würzburg. So the idea is that you make a country, like a developing country where there's an outbreak, to produce a vaccine itself.
Yes.
And you showed that in a clinical trial in Mexico. But here they produced so you said 4,500 vaccines for 4,500 people. But at the end, this is nothing. So when there's an outbreak, I mean, how many shots are produced? 1 million,
Yeah.
several million? Is it possible for a developing country to produce so many?
Yes. So the reason it was only 4,500 is because we were doing a clinical trial. But even if you have an outbreak, you have to do some testing. You can't just go and make a lot of vaccine unless you know everything about it beforehand. So what this example shows you is that with a very small facility that we have, where we made only 4,500 shots for the clinical trial, we have the capacity to make millions of doses using the equipment that we have. So that same facility, if we had an order, if somebody said, please give us 2 million doses, and we were ready to go, we had the appropriate...
How many people do you need for that?
Right now we have 90 people, out of which maybe about 40 people work in manufacturing. I need maybe another 15 people in order to go to the next level. So it's not like you're talking about employing thousands of people and training them and all that. This system is so simple. You can we were not ready to make millions of doses. Don't get me wrong, but it doesn't take that much to get it to a point when this facility can be producing at least 5 to 10 million doses. So you can get away from this idea that it's not just 4,500, but you can make millions of doses.
This is Zoe Weibler from the Paul Ehrlich Institute. Your virus-like particles do not need adjuvants, right?
Well, if you want to use adjuvants, it is compatible with adjuvants. What we have found is that our particles are potent enough by itself that formulation of these particles in saline or phosphate buffered saline with no adjuvant is sufficient.
Okay, virus-like particles were shown to take up parts like RNA from the producer cell line. So do you think this RNA could act like a natural adjuvant? So maybe this contributes to immunogenicity of your particles?
That's a good question. What we do is part of our quality control is look at how much host RNA or DNA is present.
Yeah.
We have found that the level of any uptake of our host cell DNA or any RNA is negligible. It's just not doing it. So the process by which this particle is assembling itself and budding out, it's not taking up any of the nucleic acids along with it. So we don't think that the nucleic acids are acting as adjuvants, but there are parts of the membrane of this particle that is coming from the host cell, and the fact that these particles contain many different epitopes of hemagglutinin, those attributes are attributes like adjuvants. So they may be self-adjuvanting, but it is not very dissimilar to how a virus actually activates the immune system.
Ingo Drexler from the Technical University of Munich. There's no recently coming up new evidence that also Tcell responses can play a major role in influenza immunology. So you have a different to have the matrix protein included, and this is a kind of conservative protein, not as mutagenic than the other
That's
two.
right.
So did you have a chance to look at these
Yes.
kind of responses?
I can show you slides outside, but I have them here where we can show that these particles are able to elicit a cell-mediated immune response. You can easily test this by taking human samples and getting the PBMCs, the peripheral blood mononuclear cells, activating them with peptide pools of the M1 protein, and demonstrating that T cells, effector T cells, memory T cells are activated through these peptide pools, indicating that yes, these particles are being picked up by antigen-presenting cells, going through the MHC1 and MHC2, both of those pathways, presenting the antigens and activating the appropriate CD8 and CD4 positive T cells.
Good argument for you to promote the vaccine, because
yes,
it's a bit
yeah,
different.
I think it is a great argument to promote the vaccines. Most people don't believe it. They don't believe it for two reasons. One, in the case of influenza, most people don't believe that you need cell-mediated immune responses to protect yourself. So even if you show data They say like, okay, well, fine. And the second thing is that you have to go through a regulatory process. And the current FDA or the European regulatory agencies, they only look at one thing to get this product approved from an effectiveness standpoint, and which is antibody against hemagglutinin. And until you can get a surrogate marker, a biomarker, that is based on some sort of a cell-mediated immune response that is linked to efficacy of the vaccine, it's very hard for us to really make a case for it. I think it's nice to show, and we have data to show that the egg-based vaccines versus our vaccine have a very big difference in the amount of cell-mediated immune response. In fact, there's a huge difference in the antibody, functional antibody against neuraminidase in our vaccine versus the egg-based vaccines. But all of that data is only it's only supportive data to then put around the hemagglutinin inhibition antibody data to say that now we have the basis of making a better vaccine.
Rahul, I have a last short question. What we face frequently is the problem of acceptance or compliance to vaccines. Do you think that in countries like India or maybe Nigeria, it would be more acceptable to have a homegrown vaccine for the population than something which is imported from Europe or the States?
Well, there are a number of factors that are important in the vaccine acceptance. I think that countries like India, where I grew up in the early part of my career, or early part of my life, people don't care about preventive medicine. Really don't. At least at that time when I was there, they don't. Because the idea is that they think about, well, we'll go to the doctor when we are sick and we'll get treated. If they have some money in the pocket, they'll spend on food, or they'll spend on clothes, or they'll buy a new car or something, but they don't think about health. I think the things like the pandemic have put a scare in people. And those are the kinds of things that trigger people to think about, I don't want to get in this mess. I want to figure out a way that I can survive this kind of thing. And so a vaccine, if I have a vaccine, I'd rather take that vaccine. So it's kind of perverse, but things like the pandemic are things that will increase the uptake of vaccines. Media plays a huge role also in this. And you asked the question about homegrown versus international. There is obviously a soft corner that media has for homegrown companies. And so if you can have a case where you can make a vaccine that is a lot affordable from a homegrown country versus buying an imported vaccine, which is only sparingly available, I think you have a much better chance of acceptance within the country.
Thank you very much, Rahul, for this exciting talk.